Ready to start my day. So most days when I'm heading into lab, I'll bike into work. Especially on days like this where it's nice and sunny, makes it pretty enjoyable. Got ourselves locked up and yeah, time to head in. Just walked in on the board view and checked in at the front desk. We've got a good system for dealing with COVID. Put in my little my name badge and checked me in. Got a free cookie today because that's what it's like working at a kids rehab hospital. Ready to start the day at work. My coworker Allie is here. Callie, if you want to say hi. What's up? We're right now demoing a new system that we're working with a summer student, looking at kind of some rehab stuff for things you would take home after a visit to the doctor's office. Do some training with your new prosthesis. Heading into the digital technologies room. This is where I collect a lot of my motion capture data, which hopefully we'll see if I get a chance to show you guys that later. But this is kind of one of our big data collection rooms and where some of the clinicians also do a lot of their therapy. I got another coworker in. This is Harry. What's up? He's our professional engineer. We got our 3 laptops set up and right now we're trying to get the app working. And it is sort of doing that, but not really. At the CAF, it's not all work at Bloorview. We definitely take our time to relax a bit, hang out, and de-stress from the day and catch some nice sun. So all of my time's not just spent doing research. I mean, one of the main things that I came to work at this hospital is the fact that I do get to work with, the people who actually provide some of this care. And so my project is specifically focused on helping some of the physiotherapists here provide gait training to the clients here, and using things like your phone to track what you're doing. Think like step counters, but for doing some of the more complicated analysis, for really bringing data and objectivity into some of their practice. This is Kathleen. I want to say hi, and she's a physio here at Bloorview, and maybe she can talk a little bit about what you're kind of doing.
Sure. So I'm a physio, like Gabe said, up in the prosthetics department at Bloorview. I'm also working in one of the inpatient departments called Complex Continuing Care. So in prosthetics, we see kids and adults, some of them with acquired amputations, some with being born with limb absence. And we work on making sure they're walking in a way that they're able to do all the things that they want to do. And sometimes it's looking more at people who want to start running or dancing or any particular sport.
So yeah, she's gonna help. She's helping me kind of recruit some participants. I got the Extend system. This is kind of one of the systems we use for recording some of the patients walking around. So first of all, we're going to calibrate the system and actually get me into the virtual environment. So I'll have one of my coworkers who's behind the camera give me some cues for actually calibrating it.
All right, stand still.
All right, so now that I've calibrated myself into the system, you can see that when I start moving around, yeah, you can now finally get to, there I am, there's my little avatar. I don't have any arms because I didn't put any arm sensors on. That's because the research that we're looking at, really, we're not interested in that stuff yet. And so we're just seeing what happens when physiotherapists provide some of their sessions at Holland Bloorview, like what are the kind of changes that they're actually doing to some of the patients that they're seeing. This is kind of one of the prosthetic simulators we have to kind of simulate what it's like to be an amputee and kind of help us test some of the things we're collecting. And so you can see that I'm strapped in here, and this makes sure that I basically don't have any knee and ankle movement of my own. And this is something that our lab used to design. And this is kind of an example prosthetic that someone might actually be wearing. And yeah, so this is kind of something that we help to collect some of our pilot data. So our lab works with a variety of different things. You've got my project that I kind of showed. I've also brought in, you met him earlier, Harry, to kind of talk a bit about what his product is working on.
This is actually the plaster room where prosthetists and prosthetic technicians come in to shape their models. What you see here is a plaster cast of a person designed for an ankle foot orthosis. What the process will do, they'll come in and shave this model and create buildups to make it perfectly designed for the person's limb. So after they've designed their plaster cast, what they do is they come in here, heat up sheets of plastic in the oven, and mold it over these shapes, which are perfectly designed for the shape of the patient that's going to use them. What we're looking to do is involve technology like 3D scanners and 3D printing to really reduce the amount of waste and time that the prosthetist spent designing these devices, and at the end, hopefully making a product that's better for the patient.
So I hope you enjoyed this glimpse into kind of a day in the life of a research student here at Holland Bloorview. It's really awesome to be surrounded by people working on working with everyone from amputees to people with cerebral palsy, really anyone in between, to really just help people live the best life that they can. And it's what keeps me coming back, really getting to work on research that's pushing the cutting edge of rehabilitation and health. And so now it's time for me to head home and and then restart the day tomorrow.
Hi there, nice to meet you. My name is Katya and I'm a third-year PhD student at the University of Toronto. The work that I do involves generating tissues in a lab, and that is basically we study and learn how to build human tissues, particularly the ones surrounding the heart, in a lab setting. So today I'd like to take you on a day in my life, and before we head out to the lab, I'm just going to grab a quick breakfast and then we'll walk on over to Mars, which is that building right there. Alright, so here we go. We're at the Ted Rogers Center for Heart Research. And I'll head in and gear up and I'll see you inside. So for us to build tissues in a lab, we need to very deeply understand them. And one way of doing this is to reverse engineer the tissue. And what that means is to take the tissue and break it apart into different components and understand how each of them works. You can understand this on a biological level, on a chemical level, or even on a physical level in some cases. Since I'm working on pulmonary arteries, I'm mostly interested in the biochemical side of things, as well as physically how they're able to take on loads. And this is mainly blood pressure, for example, and how they're able to adjust with those loads. So what we're going to do today is run some histology. I have some pulmonary artery samples, and I'm going to freeze them down in OCT, take them to a cryosection, which is a machine that's able to slice them into very thin slices, and then take those and stain them with dyes so that we can, at the end of the day, figure out the different components in that tissue. So to run tests on tissues, one thing we can do is to preserve them. And there are two main ways of preserving tissues. One is through putting it or embedding it in a wax. A very common one is paraffin. And another way you could do it is to freeze them down, kind of like you would freeze your food in a fridge in order to protect it from getting spoiled. And we do that with this compound called OCT. It's kind of like a gel. So I have some OCT here in this little container, and I do have my tissue as well, this little sliver of tissue. And I'm going to place my tissue in the OCT. And we want to avoid any bubbles in there because once that freezes down, it can actually damage the tissue. So we're going to fill that up a bit more. Make sure that the tissue is nice and protected. And then we're going to drop it in liquid nitrogen. So for nitrogen to be in a liquid state, it actually needs to be at about -200 degrees Celsius. And that's what I have here. I have a little canister of liquid nitrogen. So if you can imagine, this tissue is going from a fridge at around 4 degrees to being dropped into something that is -200 degrees Celsius. And that's going to immediately freeze the tissues. So what it does is it stops everything immediately at the state that it's in, and that's a very good way for us to preserve what's going on inside the tissue.
Taking a look at the inner workings of a cryosection, we see that the frozen block of OCT, with our tissue at the center. There's a sharp blade behind the metal platform that slices through the block when it passes over it, creating a thin sheet that's about 20 microns thick, or about 1/4 of a strand of hair. Everything inside the cryosection is constantly maintained at -20 degrees Celsius, so we can carefully unravel that thin frozen sheet. And when we bring in a glass slide that's sitting at room temperature, the sample will instantly attach onto the slide and the OCT will melt away.
So after we've frozen and sectioned the samples, we can take them into the lab and run histology on them. And what histology is a process of staining your sample with specific types of reagents and dyes. And it can be a pretty long process. It all happens in the dark because most of these reagents are light sensitive. But what we end up with at the end of maybe a whole day of work is some very vivid pictures of your samples with different colors staining for the different properties in the sample. So whether you have a certain type of fat or a muscle or a type of cell specifically, and it makes for a very interesting presentation. So it's been a few hours since we stained the tissues, and it's finally time to take a look at the images. To give you a quick recap, what we're looking at here is the same slivers of tissue that I froze down and stained, and they're on this glass slide. So we're going to take the slides over to this lovely optical or light microscope, and placing it on the stage, we can use these lenses to magnify it to about 40 times its original size. And that's what we're looking at here. So this is the pulmonary artery vessel wall, and the wall itself, this is the inner side of it, called the lumen. So blood would be flowing through the vessel on this side, and on this side would be any of the neighboring tissues. And as we can see with the staining, these black lines here indicate that there is layers of elastin in the tissue, and elastin is a protein that provides these vessels with their elastic recoil properties. So when they're overpressurized, they can change their size. This little bit of blue that we're seeing here is a stain for a mucopolysaccharide, or proteins with sugars. And on this side, the bit of yellow that we see is actually a stain for collagen. Lastly, these little purple spots that we're seeing everywhere, well, those are the cells. And kind of understanding the thickness of each of these layers, as well as the distribution of the cells and the cell morphology, so the shapes of the cells, we can get a sense of whether or not this tissue is healthy, or if it's diseased, or if it might be somewhere in that transition between the two. So thanks for coming with me to the lab today. I hope you were able to see something new and exciting. The Ted Rogers Center, just like countless of other research facilities on campus, is always filled with scientists, students, and clinicians who are trying to work towards a common goal, and ours is to fight heart failure. No 2 days in the lab are ever exactly the same, and so I can guarantee you that if you have a curious mind, there are still thousands of questions waiting to be solved by someone like you. If you have any questions at all, don't hesitate to reach out to me. And also, across Ontario, there are tons of amazing summer programs for research at schools and universities. That would be a great place to get started. So thank you for watching, and good luck with everything.
Chipper!
Good boy. Good boy. Let's go swimming. Come on. You can do it, Chip. You can do it. Yes, Chippy. Do it. Yeah.
Okay, so here I am in the microscope room. You can see my microscope here. We actually borrowed this microscope from another lab, which is really nice because ours is currently broken. And so basically I'm going to be here for 4 hours imaging 4 different cell culture plates for my experiments. And then in the afternoon we're going to prep some blood that we actually get from donors who have donated their blood for research, which is something that a lot of people do, which is really nice because it makes our research possible. And we're going to be extracting 2 different kinds of cells from this blood, which comes from all the way in Vancouver, British Columbia. So they ship the blood at room temperature, and then we use it the next day. So that will arrive, and I'll introduce you to some other researchers who are working on that type of blood as well. Okay, so here is the microscope that we use, and here is the cell culture plate. So the blue light is basically where our dye will get illuminated, and then it will fluoresce, and the detector will read that out as our image. So here is our image. I just put it in black and white, but what you're actually looking at are green cells. So basically the image is going further and further into the gel, or you could think of it as further and further into some jello. So you can see that these cells are far in the gel, and this will differ depending on the treatment of the cells, and that's the type of experiment that we do. Okay, this is Amber. Amber, who are you?
Who am I? Hi,
Yeah, who are you?
I'm Amber.
But this is very common in science when you're delayed, right?
Uncontrollable.
So now we're gonna eat lunch. So fun.
Yeah, some lunch.
Okay, so the first step in doing the blood prep is to sort out the cells by density. So we have this special solution in here, which is called Phycol. And the red blood cells, after we spin the blood, are gonna settle to the bottom, and the white blood cells are gonna go on the top. So I'll show you what Amber's doing. So here Amber is using a big pipette in the sterile cabinet so that she's protecting the blood from contamination. You can see the blood is sitting on top of that solution, and then we're gonna spin it really fast, and the cells will separate based on their density. That's right, Amber? Yep. Okay, so here is the blood, which has all separated. So on the bottom, you can see the dark spot is the red blood cells. And then in the middle was that material that we used to separate. And on the top, you can see a layer of the white blood cells. Maybe this one will be a little bit clearer. It's a very thin layer. And then on top is all of the different proteins within the blood. That's what that kind of milky area looks like. So the number on the top of mine is 91, and on Amber's is 90. That means that we have processed that many donors. Can you believe it, Amber?
Not really.
After 2 years, right?
Yep.
Okay, we'll do the next step, which is transferring the white blood cells to a tube, and we'll spin them down. So fun! Yay! Okay, so now what we're gonna do is sort our cells using a magnet. So you can see here we have these little columns, and basically what's going to happen is when we insert them into this magnet and we flow the cells on top, the cells are going to stick into the magnet and the rest of the cells are going to come out the bottom. Okay, so here are the cells and they're going through the column, and a lot of them are coming out of the column, but some of them are sticking to this part of the column. And then once we flow all the cells through, then we're going to elute the cells that we want so that we can culture them on our hydrogels. Nice.
Oh, that's Kristen. She's helping me today.
Oh, hi, Gracie.
Hi.
I am trying to take a vlog for students to show them how does this work in the hospital. How does the data collection? It is the data collection.
Yeah.
We're going to take
data collection. So I'm gonna put a lot of this gel on your head. No, not a lot, but try my best to not put as much.
What is this gel for?
To make
EEG?
to improve the conductivity of the EEG electrodes with your skin.
Can you explain EEG briefly, just in 1 or 2 sentences?
Yeah, sure. EEG is just a couple of electrodes we put on your head, and it will measure the your brain activity. So electrical activity from your brain, it kind of reaches the surface, and we can pick them up with those electrodes, like surface electrodes. They are very noisy, but we pick something up, and if we filter them and all of that good stuff, we can detect some meaningful information.
You put the EEG cap on my head?
Yes, that's the EEG cap on your head. We are putting gel on it to make all of these that you see. If they're red, that means the conductivity with skin is not good. They are not detecting your brain waves. So we put in gel and make all of these green, and when they're green, we detect. It means that it's reading something.
Okay, great. How do you feel right now? It is so interesting. Now you're filling it with gel?
Yep. Yeah.
Cool.
And then we put this on. It's blunt, so
yeah.
nothing will happen. In front of you, 2 circles will appear. After a few seconds, a white cross will appear. Always keep your gaze at this cross. Then one of the circles will flash quickly as you are focusing on this cross. Focus or you'll miss it. The flash will be quick. If the right circle flashes, squeeze the ball in your right hand. If the left circle flashes, squeeze the ball in your left hand. Squeeze as hard as you can. This is shown in this video. This is the EEG signals.
I finished the test.
Yeah. Oh, you finished it? Thank you very much for participating in research. There's nothing really obvious from all of this. Just have to filter them a lot and average them across a lot of trials. Do you want to blink? You can take the blinks. Oh, see, here is her blinks. Down here, she's blinking.